And we talked about this movement of solutes. In this case, these little black hexagons, right, are highly concentrated and they're due to their kinetic energy, right, or the blue dye crystals over here. They're moving all around until they come to dynamic equilibrium. Right? So this is the fusion of these solutes down a concentration gradient, right? And they're doing it in water. The thing is about this movement is that it's not just these things that are moving this way, right? The solvent, in our case, it's always, we're talking about water here, is also going to undergo diffusion, right? So let's just say when you put a drop of blue dye here, it displaces water, right? So all of a sudden your water concentration has become decreased, right? So what's going to happen to these water molecules? They're going to move, right, toward their low concentration, which is toward the area of the solute, right? Because this has displaced the water right here. So at the same time that this solute is moving toward down its concentration gradient, the water molecules themselves are going to be doing the same thing in the opposite direction, right? Everybody clear on that? That's there's a big kind of concept here, right? Just to kind of conceptualize. These two things are going on at once, right? For all these blue dye molecules that are kind of spreading out this way, there is also water moving in toward there, right? For our purposes, if you're taking chemistry class, I'd say no, but in physiology, yeah, we're talking about very limited substances here. We're always talking about water, and 90% of the time we'll be talking about, no more than 5 molecules, sodium, potassium, glucose, bicarb acid or something like that, right? So it's pretty limited. We want to stick to the physiology part of it, human physiology part of it. So it's always, for our purposes, always going to be water, the solvent. All right, so why am I talking about this water movement? Because we get to our everybody's favorite subject, osmosis. Right? This is defined as the diffusion of water molecules, right? Diffusion of water across the cell membrane, not just in a glass of blue dye. Right? It's across the cell membrane, right? Whether that's through the lipid bilayer in limited fashion or through these channels, these water selective pores right here, where it can diffuse down its concentration gradient. And by its concentration gradient, it's going to be going where there's less water molecules, and usually there's less water molecules because there's more solute particles, right? So whatever the solute diffusion might be, osmosis is usually in the opposite direction of any particular solvent, solute, and hence follows the expression water follows salt, right? And the proper term is water follows solute, but doesn't water follows salt sound a lot better? But it means just water is following, right, whatever the highest concentration of these solutes are or salt is. Everybody clear on just the basic definition of what's going on with osmosis, right? Diffusion of water across the cell membrane, not just in a jar of dye or something like that. It's across the cell membrane, and it's going toward its low concentration. And by that, we mean the opposite of what the solute diffusion would be. So in this situation, if we want more water to come in here, we'd add more salt. If we want more water to go this way, we'd add more salt and the water would follow, right? This is an important point too, right? How we control water balance in our body. All right. So here is the artificial kind of setup that is often used to describe what's going on with osmosis, right? They have this U-shaped jar right here in the middle. At the apex here, they have this selectively permeable membrane. That's the cell membrane, right? And then this is, let's just say this is the intracellular fluid and that's the extracellular fluid, right? But this, for some reason, is easier to understand. But that's the setup. This is the kind of model they often use for this. All right, but in this cartoon here, you have this selectively permeable membrane. These little green dots cannot get through this membrane. So at a starting point, we have 6 green sugar molecules over here, and let's just say 12 over here. Right? So you have that and that. There's an imbalance, right? The water, in that case, even though it's moving back and forth, there'll be a net movement of water toward the area of high concentration of fluid, right? These molecules can't get through here either way. The water molecules can, so they're going to follow the sugar in this case. They're going to move toward there as a net movement sort of thing, right? There's movement back and forth, but the net movement, the osmosis, is occurring this way, right, toward here. So what happens is that the water keeps moving that way, and it moves so much, right, that it moves until the concentration of the solutes is the same, right? So here you had a cup of water with 6 molecules. Here you had a cup of water with 12 molecules. Now you have 2 cups of water with 12 molecules, which means that there's 6 molecules per cup. Okay, so, and this is 6 molecules per cup. Right? So the concentration is the same. The water was driven toward that higher concentration of sugar until it's equal. Now there's an equal number of water molecules on each side. Even if it doesn't seem like, seems like there's more water, they're equal because the sugar is taking up that space, hypothetically, right? And it's done it so much, right, that it's pushed up, right? It's going up against what seems like, why would it be going above gravity, right? We all know water kind of follows the easiest path, right? This is all set apart by gravity, but it's going kind of pushing up against gravity. The movement is such that it kind of creates a little force here. So this movement over here, this osmotic pressure, this movement over here, right, toward an area of higher solutes, the solution over here is said to be creating this osmotic pressure that the water is going to move toward, right? So when you say something has a higher osmotic pressure, then the water is going to be kind of sucked over there, right? It's a pressure, but the water is going to move toward that pressure. Right? So that's a kind of weird concept, but that's the concept of osmotic pressure, right? This is exerting osmotic pressure, right? And the water is drawn toward it. So it does it, it keeps on doing it until the osmotic pressure. So the question is over here, you started off with this much water, and then it's gone down, and this has risen a little bit here. Why doesn't it just keep going until it flows over the side? Right? And at this point, they're saying it's reached equilibrium, right? The osmotic pressure has stopped, and for whatever reason, there's no net movement anymore once it reaches this height right here. Okay. So why doesn't it keep going? There's another force to make things complicated called the hydrostatic pressure. In this case, in this little experiment, it's really gravity that's they're both keeping the water down right here. The osmotic pressure moves it up until the osmotic pressure is only so much that it stops, like it can't overcome the gravity. So it stops right there, right? So that's the hydrostatic pressure. In this case, could be gravity, but this could also be blood pressure or something like that, right? A pressure that's opposing that osmotic pressure right there. So, and here's the kind of example we're going to try to understand here. Selectively permeable membrane, molecules on one side, osmosis is going to occur in this direction, right? And it's going to do it, right? It's going to do it until it reaches a certain point, until it stops, right? But it stops at some point. Then the way they measure this is they say, well, if we applied a pressure that brings it back to this position, right? How much pressure would we have to use? That's the applied pressure, and that's going to be equal to the osmotic pressure that this force is creating right here, right? And so if you don't want to follow that, that's fine. Just know that this right here is creating this osmotic pressure right here. Right? It's exerting a force on this side of the water. That is, it's being kind of sucking the water on this side. and whatever's in that water. So here's a couple of examples. And I kind of mentioned this when I talked about mixtures and colloids, right? Here's the blood pressure coming in through a capillary network. There's a capillary pressure due from this is basically one of those hydrostatic ones, and it's got this amount of force, right? Measured in this right here, right? This is all due to blood pressure. And movement is this way because the interstitial fluid pressure is much lower, right? So stuff is going to move in toward here. Stuff is going to be pushed out from the capillary pressure. Things also going to move in a little bit this way. As you move along here and the capillary pressure becomes less and less, you have more concentrated proteins and stuff. That's going to exert a osmotic pressure called the colloid osmotic pressure. That's going to draw stuff in over here, right? So that's going to draw like fluid and stuff into this, right? And that's how you get rid of, you bring in good stuff over here and you bring out waste based on this osmotic pressure that's created, and stuff is going to get sucked into the blood vessels at this point. And this is all due to solute differences right here. This is due to that hydrostatic pressure. Here's another example. Over here, these are the kidney tubules, which you'll be tortured in Bio 145. It's a very complicated kind of setup where you have these tubules running through the kidneys. And over here, it's kind of the interstitial fluid and the tubular fluid. This is your filtrate that you're trying to pee out, but it's got a lot of water that you want to reabsorb. So it's equal, the osmotic pressure, there's no osmotic pressure. And then as you dip deep down into the kidneys, this pressure gets more and more. That is, the solute concentration gets higher and higher, right? And water flows out accordingly, right? Toward that high salt concentration, right? Because it's really salty here, the water wants to move toward that salt. And this is the way we reabsorb water, right? In certain areas of our kidneys, right? And so the other thing I want to bring up about while I'm at this example, says urine has this low volume of water, has a high osmolarity. That is, there's a high solute concentration because we've reabsorbed a lot of the water right here. And so this term osmolarity is going to come up in a few contexts. I just want to go over it. I won't ask you a specific question on it, but you're going to see it over and over. And it's good to know. If you've taken chemistry, you know about molarity. And this is osmolarity. So what this does, and it's not so important to know, except to know that, or maybe it will be in Bio 145, but you'll see the term milliosmoles and stuff like that, right? And what it does is take into account that a sodium chloride molecule has a certain molarity over here, right? But when you put it in solution, which is always going to be, it dissociates into sodium and chloride. Each one of those particles is going to exert its independent force as a solute. So now instead of one molecule, you have two molecules. So 1 mole of sodium chloride is equal to 2 osmoles of sodium chloride, or osmolarity, right? Glucose, which doesn't dissociate, there's no change in that. 1 mole of glucose, 1 molar glucose is 1 osmolarity of glucose, right? Same thing. If you have 3 molecules, it's going to be 3 times because they dissociated into calcium and 2 chloride molecules. So that's just the whole concept of osmolarity, right? You'll see it. I showed you this sheet before. When you're giving somebody IV, you want to know how much to put in some, it's up here. It's M, it stands for milli, but milliosmoles per liter. Right? You'll notice, it also has it in the grams per liter as well. You'll notice you double the dose here, you get a double in osmolarity over here, right? And so glucose, if you looked at the actual molarity, it wouldn't change anything, right? For over here, for sodium chloride, notice that it's a 77 millimolar. 77 times 2 is something like 154 osmolarity, because these broken up into sodium and chloride over here, right? This 154 millimolars turns into 308 milliosmolarities, So those are just because each one is considered independently, right? All right, so I want to bring that up just because it does, you'll see that term milliosmoles and stuff like that. It's just the fact that these ions, these salts and acids and stuff break apart in solution, and each individual molecule is going to exert its own little force. So osmotic pressure, right? That's the pressure created by the amount of solute versus, in one kind of area versus another. In our context, it'll be inside a cell and outside a cell, right? Isotonic solution means that the solute concentration of this is the same as the red blood cell inside, right? That is the same levels of solutes. Even if the solutes are different, there's no osmotic pressure in an isotonic solution. And so when you use the tonicity is a measure of the solution's ability to change the volume of cells by altering their water content. So we talk about tonicity, right? And it's related to the concept of osmolarity. And we always use it in reference to mostly the extracellular fluid, not the cell itself, right? In this case, there's no pressure either way. So whatever the overall osmolarity, they're equal. There's no net movement. The cell is nice and healthy, right? Osmotic pressure 0. If we put a solution in here where there's less solutes in this solution, that is, would be the extracellular fluid, than there is inside the cell, what would happen, right? Osmotic pressure is going to drive water inside the cell, right? That's going to cause problems immediately just with dilution stuff, but eventually it's also, if it's strong enough, it'll also burst the cell, right? So that's a hypotonic solution. That is, the solution has less solutes than the cell, right? So when we say hypotonic and everything, we're comparing the solution to the cell. So the water is going to follow wherever the solute is. Here, there was higher concentration of whatever, water's following it into the cell bursts. The opposite situation is if you put a very salty solution here, you put a normal cell and say, I don't know, salt water or whatever, right? There's more solutes out here, and the water leaves the cell to try to dilute that water, right? And it'll keep on doing it. And it's called crenation. The cell crenates, right? That is, most of the water is going to leave it, right? Osmotic pressure is going to drive water outside the cell, right? So water is going to follow the solute there, right? So that's just what is happening with these isotonic, hypertonic solutions over here, right? And it has to do with the osmotic pressure that's created by a difference in, the solute concentration in the cell and outside the cell, basically the intercellular fluid and the extracellular fluid around it. Right? That's tonicity. So next little topic is this concept of fluid compartments, which we went over on the first day when we went over general body organization. We talked about these fluid compartments, the fact that we're made up of, 50, 65% water or whatever. And the rest were whatever else solids. And within that fluid over there, right, it wasn't just sloshing all around. It was in these kind of compartments in some way, right? It was either in the cells themselves, right, making up most of it over here, or it was in the extracellular fluid, inside the cell or outside the cell. Outside the cell could mean either the water surrounding the cell or in the blood or a few other places right there. And so there's these so-called fluid compartments over here. We're going to kind of talk about fluid exchange between these and, how this relates to a lot of the physiological stuff that you're going to be covering later on. Right? So as far as these fluid compartments, your plasma, right, the blood, right, this looks like a lot, right? So all of your blood vessels, right, hold all your plasma, which is mostly water, right, and it's flowing all around your body right there. So that's your plasma, and then there's a few other little things like your cerebral spinal fluid, your joint cavity stuff, whatever, right? But that's one little part of your extracellular fluid, right? And then for your intercellular fluid, it was all about the fluid, basically the cytosol inside the individual 70, 100 trillion, whatever cells of your body, right? So it's weird to call it a compartment. It's like 100 trillion little compartments that hold all this water in there, all the cells of your body. You could think of a piece of nice juicy steak versus a piece of beef jerky to get an idea of the fluid, right? What the fluid is do for you. And then the rest, right? That was the intercellular fluid. The rest was this interstitial fluid, right? The fluid that's surrounding, right, the cells themselves, right? The fluid that's surrounding the cells themselves. And for this, remember just to put it into a concrete context again, right? The blood vessels are easy to understand. Inside the cells are easy to understand because you could see them, right? But each one of these cells that make up the epithelial lining of the gut tube, all the fibroblasts in here, all the glands, everything like that, right? They all have their little intracellular fluid, but there's some kind of interstitial fluid between it, especially, a lot is held in that connective tissue around here, right? But even between these individual smooth muscle cells that make up this layer, there's going to be some kind of fluid in there, right? To bring all that oxygen and glucose and stuff like that. Right, so your interstitial fluid is going to be surrounding each of these cells, even if it doesn't look like it in this picture. Right, just keep that. They're not a bunch of cells floating around like amoebas in a pond, right? That you would think if you took this picture literally. Again, here's another concrete example. Here's an individual muscle cell, right? That could be as long as your, a foot long or whatever. And each cell is going to be surrounded by a little fluid, capillaries, but there'll be also a little bit of interstitial fluid around the cell, right? So this is this cell right here, even though it doesn't look like it. And there's going to be surrounded by some kind of fluid. That's the only way that oxygen and glucose and stuff is going to get from the blood vessels into the cell itself. All right, interstitial fluid. And so as far as these, right, your cells, your interstitial fluid, and the plasma. And this is the basic relationship of them, right? Your cells are surrounded by interstitial fluid, and your, the plasma, the blood is separate, right? And it's going to deal with the interstitial fluid first, right? So what we're going to see is that, stuff is going to come out here. It's going to go in the interstitial fluid, and then it's going to deal with the cells right here. The plasma stuff is not directly connected to the cells. You got this intermediate kind of space around which stuff is going to diffuse through. So for this first exchange, this has a lot of words. Don't worry about any of these details. You'll cover this. Maybe we'll cover a little bit in the nervous system, but basically your capillaries, right, which are running through your body's tissues. Over here, they're lined by these endothelial cells. And, there's going to be diffusion of stuff through the individual cells. So you have a little bit of control. And you also have these pores, like spaces in between them, either small ones or big ones, depending on where you are. Right, so there's a pretty free exchange of fluid between the capillaries and the interstitial fluid right here, right? Whatever can get out of these holes right there is going to go out, and it's going to be driven by like hydrostatic pressure, right, on the one end, and maybe osmotic pressure on the other end, right? The fluid exchange here, and then whatever kind of more control you might have by going through the cell membrane, right? So the capillary interstitial fluid exchange is pretty free, right? We don't have that very tight control like we do, and we'll see when we get to the actual cells themselves. Right? So this is the extracellular fluid kind of going on here, and they're kind of equal, right? So here's your blood plasma, right? And it's going to exchange first with the interstitial fluid. The interstitial fluid is going to exchange with the capillaries. And then the cells are going to do it with the exchange with the interstitial fluid and vice versa. Okay, so these are these kind of compartments. This is what's going on. And we saw that there's a pretty free exchange between the blood and the interstitial fluid. Big things like proteins and stuff aren't coming out, but everything else is pretty freely shared, right? So the makeup of this and this are going to be very similar. This is just the same picture, right? Water, sodium, all the stuff in here. And here is going to be mostly the same with the exception of these bigger proteins, which can't leave the capillaries right here. And then the one place, of course, that the interstitial fluid does not separate the blood from the cells is the blood cells themselves within the blood. However, the compartments idea still holds. The intercellular fluid of the blood cells is different from the plasma of the blood. But otherwise, if you look at the composition of any particular solute, if you look over here in this chart, blue and red are the extracellular fluid, interstitial, and plasma right here. You'll see sodium is about the same, chloride is about the same, bicarb ions about the same, potassium ions about the same, red and blue here. Right? All these are about the same, with the exception of the proteins, right, which are going to be much higher. You have all these blood proteins that aren't in the interstitial fluid. But all these kind of electrolyte type of stuff is going to be about the same in between these, right? And the difference is, here's the intracellular fluid, the cells, right? You'll see something like, here's the level of sodium inside a cell versus the extracellular. Here's the level of potassium right here, right? So there's a big difference in between, what's in between the extracellular fluid, and that'll include the free exchange between these, and then the extracellular fluid and the intracellular fluid. Right? So you have this selectively permeable membrane between the cells. This should say interstitial fluid. This is one of the problems with ICF, meaning interstitial and intracellular. Interstitial cells and ECF, it should be, set up these electrochemical gradients, right? Because they're going to have more sodium out here, less in here, potassium, vice versa. But overall, right, despite all that, they're going to have about the same osmolarity, right? So there isn't any of that hypertonic, hypotonic when everything's going well. You want that normal isotonic, your red blood cell or any cell to be in an isotonic solution. The difference here between here is going to be like those different molecules, they're going to be distributed differently, but the overall osmolarity, that is the osmotic pressure, should be, if everything's working right, should be close to zero, right? All right, so what happens? What happens when there's fluid imbalances, right? Well, this is usually due to change in some kind of, in the osmolarity of extracellular fluid, whether it's in the blood or in the interstitial fluid. This causes a whole bunch of problems, right? Ultimately, we're leading to one of these conditions, right? If your blood is going to be hypotonic, right? Too much water, right? It's going to end up equilibrizing with your cells, so that the cells kind of, the blood is going to match the interstitial fluid, and then the cells will try to match that, right? And vice versa if your blood is too salty or whatever, right? So these changes in osmolarity, that is the solute concentration, the osmolarity is going to cause these imbalances, right? Which can cause a lot of problems. So, example, in this case of excessive blood loss or excessive sweating, vomiting, or diarrhea, and then you become dehydrated. I mean, what happens when you become dehydrated? You want to drink a lot of water. If it's plain water, you're going to water down your blood, right? And then you're going to have decreased sodium concentration, a condition called hyponatremia. The extracellular fluid, which is going to consist of the plasma first and then the interstitial fluid, right? And so when you have this decreased osmolarity, what's going to happen around it, right? You're in this hypotonic solution, right? So water is going to go toward its concentration gradient into the cell right there, right? So the osmosis from the extracellular fluid into the intracellular fluid, and maybe going hemolysis, or maybe some kind of just watering down of what's going on in there, right? So that's a big problem, right? And it's all happening because you've lost a lot of your solutes in these conditions, right? And that's going to lead down to your cells eventually, right? Not just this feeling of thirst that you have, right? It's a real problem. So what's the opposite situation? If you have too high osmolarity of your blood, that's going to result eventually in an equilibrium with the interstitial fluid. And then you're going to have high concentration around there, which is going to cause water to leave the cell, right? More problems, right? Because you want that cell to have the nice water balance in here for all its activity, right? And it's going to undergo crenation, right? This is a red blood cell, but this would be anywhere in your body. So this brings us into the regulation, right, of this. In this case, this where our blood has gone hypertonic, right? You have increased blood osmolarity, right? When you do that, it's because you have less water, so your blood volume is decreased, right? Which decreases your blood pressure. All these are sensed, right? These are the disruptors, the stimuli or disruptors, right? Basically increased blood osmolarity, right? They're going to be detected by these different kind of receptors, right? Whether it's in the hypothalamus or all these other ones right here, right? Doing that, and a feeling of your dry mouth. All this information is going to be sent to your control center. In this case, the hypothalamus. Don't worry about the details right here. Right? And what's that going to do? It's going to have you make you feel thirsty, right? Basically give you that sensation of being thirsty and drive your behavior to increase your water intake, right? When you do that, your blood osmolarity decreases, right? You've watered down your blood a little bit. That increases your blood pressure. And, we've gone back toward lowering all the initial disruptors, right? Homeostasis. Right? Everything we go through, we're going to go over this again and again. So that's like a behavioral thing that to balance water, right? We've got less water in us. Our osmolarity has gone up. So we just drink water to fix that problem. Other things, right? Water reabsorption. In the kidneys, for instance, your kidneys, one of the main functions here is to not only get rid of like waste, but to kind of balance your water, right? So what happens, again, you're going to learn all about this. Don't worry about the details. But basically, this is a great combination of anatomy and physiology right here, right? This is where one of the biggest overlaps actually is. Right? So in your kidneys, you got these little pyramids right here. Don't worry about the details. I'm not going to ask you any of this. But in these kidneys, these pyramid-shaped things, right, you go from this isotonic extracellular fluid around here. And as you move down here, right, you can see these numbers right here. These are your milliosmoles. Over here, it gets very, very salty down at the tip here, right? So as fluid is moving through here, and this is permeable to water, water is following the salt over here to reabsorb the water, right? Because you don't want to pee out all your water. You want to reabsorb it back into the body right here, right? And there'll be a system of blood vessels to pick all that up, right? So water is following the solute is the basic key right here. And this is the way we reabsorb water, right? Using this sort of relationship between water and salt. Here's another one right here. Over here in this other part, we've reabsorbed most of the water, but if we need to reabsorb more, hormones come in and put in these sodium channels, right? We increase the absorption of sodium that's in the filtrate. What happens when you increase the reabsorption of sodium? Water is going to follow that sodium, right? Because water follows the salt over there, right? So we're using this. We're not taking water in this point, right? We're just bringing in sodium, and this is under hormonal control, right? When we decide we need more water, but the water is going to follow the salt. Here's another one. So this is all your kidney stuff. Again, here's another homeostatic regulation. Again, here is the stimuli or disruptor, decrease the same ones we saw before, right? And these are detected by something, a receptor. What's the action? In the hypothalamus, you increase synthesis of ADH, antidiuretic hormone, vasopressin. Don't worry about the details. That's the that's the control center, and this is the effector, which is going to go into the kidney, and it's going to add a bunch of water channels in that last part of the kidney over here, right? Just like I said before, you want to increase the permeability of the water channels. So what the action of that hormone did was add water channels. So you're increasing the permeability. Water is going to go out here because remember, this is a super, super salty kind of concentration area right here. So water is going to be more reabsorption. Right? So when your body needs to reabsorb more water, what do? You add more water channels by this hormone right here. But you're doing it based on that idea. Water is going to follow the salt right there. Right? This is another reason why when you drink alcohol and stuff like that, or coffee. They are diuretics, right? They kind of block ADH and they block this formation. So you pee out a lot more watery pee. So water follows salt, right? And here's that same picture. I don't know why I added this in here, but homeostatic regulation, right? Osmolarity of plasma fluid detected by receptors. ADH goes out, increases water reabsorption until we have more water. All right. So I think that covered all those objectives that are there about water kind of exchange between the fluids, and then you understand it on the basis of water following salt often, right? And the exchange between the blood vessels and the interstitial, the selective permeability of the cell membrane, et cetera, diffusion and all.